Good afternoon, everyone, and welcome to spring. It certainly feels like spring, and everybody's out there. But we're going to be inside for about a couple hours so that we can learn a lot about turf and turf diseases and pests. And with that comes our first speaker, Dr. Albrecht Koppenhofer from Rutgers University. He received his graduate degrees in biology and agricultural sciences. He was a visiting scientist and postdoc in the Department of Nematology at University of California in Davis, where he studied ecology and application of entomopathogenic nematodes and biological control of white grubs. Since 1999, he has served in his current position as researcher and extension specialist at Rutgers. His program centers on developing IPM for white grub complex, annual bluegrass weevil, and black cutworm in turf. Dr. Koppenhofer also has studied biological control, mating disruption, efficacy, and non-target effects of new chemistries, monitoring methods, insecticide resistance. He has authored over 100 refereed scientific publications and numerous book chapters. He received a recognition award from the New Jersey Turfgrass Association in 2015. Welcome, Albrecht. The floor is yours.
Thank you very much. Good afternoon, everybody. I'm going to share my screen here. Okay, so I'm going to be talking about these nematodes today. So insect-pathogenic or entomopathogenic nematodes. And as such, long word, little time, I'll call them EPNs. And a little introduction on these EPNs. So I've spent a lot of time on these, especially in the earlier parts. So nowadays I have to do all kinds of things, of course, but yeah, I still do some research on these nematodes. So they're obligate lethal parasites of insects. You find them in nature everywhere, except for Antarctica. They have been found so far. And they come in a package with bacteria, symbiotic bacteria. Each nematode species has its own bacterium. And some people compare them to, let's say, like a rocket being a nematode that moves around, and the bacteria being the warhead, which kill the insect. But it's more complicated than that. They work together. To simplify it, you can look at it that way. And there might be thousands of species of those if you look everywhere. So far, more than 26 species in the genus Heterorhabditis have been described, and more than 100 species in the genus Steinernema. They have the ability to search for hosts, anywhere ranging, depending on the species, from ambushers, which sit and wait like a spider in a web almost, and don't move around a lot, but they can do that too, to other species at the other extreme, which are cruisers. So as the name says, they cruise around more, they're more actively searching and will be more active throughout the soil profile. The host range varies. Some species like Steinernema carpocapsa, we'll mention that today. In the lab, you can kill hundreds and probably even thousands of insect species if you bother to test. But then in the field, things get a bit more specific, and under natural conditions, probably even more. Other species are rather specific to a certain type. Like there's one species that seems to be rather specialized to mole crickets, and another one we'll mention today to scarab larvae, white grubs. And these things can be mass produced in fermenters outside of insects. And of course, like most biocontrol agents, they can have the ability to recycle in the environment. The picture you see here, those are infective juvenile. That's a juvenile stage, which is the only one that can survive outside of an insect and is the one that you would find in a product that you would apply. Here's the life cycle of these nematodes in general. So you see on top, again, these infective juveniles in the soil. They're using their different strategies to find a host. If they do, they try to attach to it. They try to get inside it through natural openings, such mouth, anus, breathing openings that the insects have on most of their segments, or thin parts of the cuticle in some species. Ultimately, they want to get into the body cavity. There, they release these symbiotic bacteria, which then start reproducing. And if the host is susceptible to this combination of nematode and bacteria, it will kill it within a day or two. And that bacteria then start breaking down the host tissues, converting the insides of the insect basically into a fermenter, basically food, a broth, if you will. And the nematodes now start developing. And at this point, they actually feed on these bacteria as well as the digestive host tissues, go through up to 3 generations inside the host until the host is mostly consumed. And at that point, they will stop development again and become, again, infective juveniles that then leave the host in the hundreds to 100,000s, depending on the size of the host, and try to, continue with the cycle. Just an example here. So the bacteria actually produce pigments, and they change the color of the cadaver if the cuticle is somewhat transparent. So in these wax moth larvae, for example, that we use like a lab rats, other people would use for all kinds of things, the heterorhabditis species often turn them into, or rather the bacteria turn them into some kind of orange, reddish, or even scarlet color. Whereas the Steinernema species, they turn them often more into some version of tan to brown to almost black. In the lower left here, you see a species that I'll talk about today that we found in New Jersey about 20 years ago, new species that turns these grubs that they really like, golden yellowish to yellow brownish color. In the center down here, you see a white grub that has been killed, and you see it's orangey reddish, and it's filled at this later stage of development with all these different stages of these nematodes, juveniles and large adults. And if you remove the cuticle of that grub, that's what's on the inside there on the right, it's filled with all these different stages of these nematodes at this late stage of infection. So there is a bunch of different species that are commercialized around the world. As far as the US and what I would consider for the turf market available or making sense for that, there's just a few species. The most commonly used ones probably is on top Steinernema carpocapsae. And that tends to work quite well against various caterpillars. So the worms, if you will. So we talk about like cutworms, sod webworms, armyworms, but also billbugs and fleas. And the products, and then I'm just going to mention the relatively bigger companies. BASF is a big company, but Copper, Biologic, they're smaller companies specialized in biological control. So there's Millennium, Capson, Ecomass, just to name a few. And often they also produce nematodes for smaller companies that then repackage them and sell them under different names. Then the next species here would be Heterorhabditis bacteriophora, the one that carries bacteria. That was the first one they recognized, that now we know they all carry bacteria. And that is a species that could be used for white grubs and billbugs. And a few products here that, available would be examples would be Nemesis G, Terranem Nam, Heteromask. Then there's a relatively new species, the one I mentioned, Steinernema scarabaei, that has been commercialized in Canada for the last 5 or 6 years, and this year for the first time in the United States. And that one is a rather specific but very virulent to white grubs. And the product is called Nemagard, and the company in Canada that produces them is called One Life. And then there is one more species that at this point is really not yet used and commercialized for turfgrass, rather mostly for citrus, like citrus weevil. Steinernema Rio Brave was found in the Rio Brave Valley, hence the name. And this species should actually work quite well against various caterpillars, but possibly also billbugs and other weevils, because we found it in other systems, like in citrus weevils, and in fruit, like blueberries. We've tested it like very effective, more effective than anything else against polio. So we are planning to also test this over the next few years in turfgrass against the weevils. Like, for your interest at this point would be mostly billbugs. And that product is Nemesis R. So some basic information on how to handle these nematodes properly, because you're dealing with living organisms, and unfortunately, they don't have a true resting stage like some of the fungi and bacteria have. So they're only like somewhat dormant in the formulation, and so you have to be much more careful. And of course, they're larger than bacteria and fungi. Follow, obviously, the package instructions. They come in all kinds of formulations, granules, vermiculite, sponges, gels. And because they don't last terribly long, you should use them as soon as possible after shipping. So that's one of the biggest weaknesses of these nematodes. You can't put them on the shelf and leave them there for a couple of years, like a chemical, or even some of the bacteria, at least for a couple of years. The formulations might be stored in a refrigerator or some cooler conditions, so that it might last for a month or so. This Steinonema star B formulation, apparently they claim that it lasts at room temperature for 3 months, which is really good for these nematodes, but compared to chemicals, still not very good. The other thing is, while they survive in nature through winters, even in Canada and northern Canada even, they can adapt to that and have time with behavior and so on, physiology. You can't just take the formulation and put them in a freezer. It's going to kill them, period. End of story. Same with the heat. So they can survive in very warm environments, but under normal conditions, you just like your kids, you should not keep them in a hot vehicle in the sun when it's hot outside. That will also kill them, or leave them in a spray tank sitting on a hot day in the sun where the water can be pretty hot. So that would also not be very good. They can be applied with a lot of different application equipments, most types. The only thing is, because they're not really microscopic, they will over time, 10 minutes, 20, whatever, 30 minutes, settle all the way down to the bottom. So it won't be very well distributed. So you need to agitate them in the spray tank. Also, because they're not that small, your sprayer screen should be at least 50 mesh or coarser. Better even if you could remove the screens and just recalibrate for the application. And it can take a fair amount of pressure, but you should keep it under 200 PSI. That shouldn't be a problem. And also, they need water. We'll get into that a little bit more, but the more water you can use to apply them, the better, because you want to wash them down off the foliage of grass. So at least 2 gallons per 1,000. If you can use more, even better. Again, they don't live forever, so you should only really apply them when the target is present. They have a half-life, depending on the species and conditions, of something 3 to 6 weeks. So every 3 to 6 weeks on average. Of the nematodes that were present at the beginning, only half are left. And after another 6 weeks, only half of that. So we're down to a quarter. It's also logarithmic decline. The other thing is, they're still, even though they're not microscopic, they're still pretty small and mostly transparent. So UV light can get through in their bodies and cause some havoc, and high temperatures as well. So when you do applications because of that, you should ideally do it early in the evening or in the morning to avoid those extreme conditions. And they tend to work within a certain temperature range that varies with species, but in general, somewhere between 60 and 93 degrees Fahrenheit, with an optimum for most species somewhere around between 70 and 85. So I know my way around the times up here in the Northeast, New Jersey. So I kind of guesstimate for you guys, you probably should apply them before April and not after October, because then it just gets too cool for them to be really effective. And here is a really important thing. So these nematodes need moisture in the soil because they're essentially still aquatic. They live in the water, in the soil. So the water film that surrounds the soil particles, or that fills the voids between the particles. And so that's what they need. And ideally, moderate soil moisture. If it's completely soaked, you wouldn't apply an insecticide either, right? That wouldn't be very smart. So they can't move that well if it's completely soaked, and even less if it's very dry, because they need a water film to move through. So if the soil is dry and or hot, ideally you should apply a little water, like 1/10 of an inch or so, before you apply them. Cool down the soil, provide a little bit of moisture already. Then once you're done with the spray, if you're applying them for a surface active insect, let's say like most of the caterpillars, you want to apply about 1/10 of an inch of water afterwards to wash them off the foliage into at least the surface soil, the thatch. If you're targeting soil insects, just like with an insecticide, you want to wash that in with ideally about 1/4 inch of water to get them really a little bit down there. And finally, you want to keep the soil at moderate soil moisture for at least a week. If you can do longer, the better, because then nematodes can be more moving around more actively, effectively during that time. So yeah, moisture is a big one, very important. If you can't use the proper amount of water in your soil, then that's not your thing. That's a big limitation. There's some new research on newer chemicals. This is some older data, but just to give an idea, obviously they can be affected by pesticides. Pesticides, if they're designed to kill organisms. So a lot of them you can even tank mix, like neonicotinoids, for example. You can tank mix them. I'm sure you have done studies on that. You can mix them, for example, with acetyloprin, no problem. But there's a bunch of things, just a few examples here that you should at least not tank mix. You can apply them with separate applications on the same day, no problem, but don't tank mix them. And there are some things where you even want to separate the application timing, like chlorpyrifos, 2,4-D, trichlorpyr. You want to have at least a week between nematode and application of those materials. Otherwise, they would have a negative effect. And then obviously, nematicides, you want to give them at least 2 weeks because, well, they're designed to kill nematodes. So obviously you don't want to apply them at the same time. All right. So now I'm going to go through a couple of different insect groups that are where we have done research, or other people have done research that I summarized with these nematodes. Let's start out with white grubs. So here's a lineup of the usual suspects. In your case, in Georgia, I understand the lineup is relatively small. So you have northern half of the state Japanese beetle, and then you have different species here of these May-June beetle or Philophaga species that can cause problems. So with Japanese beetle, this is approximately the current distribution. So if you have a northern half of Georgia, you're dealing with them. And since they're relatively new, especially if they have already been there for a few years, they can be pretty nasty for a while until nature somehow catches up with them. Like in New Jersey, they're no big deal most of the time. We have other species like oriental beetle, which are more important, but they're not so catastrophic because things have adapted to feeding on them, basically. And then for the May-June beetles, because I don't know what species exactly you have, and I'm sure you have a number of species. I can't give you really information on that because we don't really even know what life cycle they might have. Here in New Jersey, they typically have a 3-year life cycle, but I assume in Georgia, they have a 2, or even in southern Georgia, probably a 1-year life cycle, which then makes them much more worse. Up here, May-June beetles rarely cause problems with turfgrass. But in the South, I know that 1-year cycle species can be pretty damaging. So here is the life cycle of Japanese beetle. This is probably a little late compared to what you guys get because it was made for more like our latitude up here. So they start out probably in your case, May, June, maybe into July, the adults are around eggs. And then they develop through until September or so into the third larval stage, only 3 larval stages. They're the ones that overwinter, come out in spring again, probably during March in your case, mostly feed for a little while and then pupate in the soil and come out and start the next generation. So you always have, they take 1 year to develop through their generation, but you have 2 generations in any given year, the ones in the previous year in spring and starting in summer, the next generation. So this is some old data, a lot of trials, and say you see the numbers inside those bars are how many trials into this summary. And that shows you that against Japanese beetle, when they were applied in late summer, early fall trials, Heteroclitus bacteriophora and other species, Deinonyma glazeri, can be quite effective if watered properly, and somewhere in the 70% control, which is just as good as Dilox. Other species like Steinonyma feltii and Steinonyma capsa, they're just not well adapted, both in behavior and ability to kill that particular type of insects. So they don't do as well. Now, of course, compared to some of the new chemistry, like the neonics or the diamides, they're better. They give you 90+% control if you do it right. So we did some research, and this is a summary of many trials where we looked at the efficacy of this commonly used species, Heterorhabditis bacteriophora, against different species of white grubs in field trials, and at two different rates. Like 1 billion is like a lower recommended rate, and then an even lower rate, 0.4 billion per acre. And you can see that once again against Japanese beetle at the full rate, we're here 78% control on average, right? But against these other species on average, we tended to see lower control rates. They're just a bit more resistant against this nematode. So when we looked into how can we improve that, and what we found, this is a lab trial, but we've seen similar things in the greenhouse or even in the field, obviously more variable. That's why I show these data. But you can see here, Japanese beetle on the left, Oriental beetle on the right, that if you apply them against first, second, third instar, the mortality caused by these two different rates of nematodes goes down. Same in Oriental beetle. And we even saw it in Oriental beetle in trials that within the third instar, the young third instars were similarly susceptible as the second ones, and so much more susceptible than the older third instar. So that means that the timing is important. And so when you do these applications, you should target ideally these younger stages, first, second stage larvae. And you do these applications earlier, you also have longer time where these nematodes then, if they kill some grubs, or a lot of them ideally, that they can reproduce and their progeny coming out can kill more grubs. And temperature shouldn't be a problem in your case because you have probably at least a month longer, warmer temperatures than we have up here. But that only does so much. So we have looked around, and other people have, and we found that there's some other species out there. And that's when we came across this completely new species that we called Steinernema scarabaei, for scarabs, because it's very virulent and rather specific to many different white grub species, but doesn't do so well against other insects, especially when it comes to naturally reproducing them. And so we found that about 20 years ago. Here's just a close-up, an early infection on the left. So they turn more tan, yellow color, golden somewhat. And then over time, the cadaver of the grub turns more yellow brownish to brownish. And I don't have field data on May beetles, May June beetles. Hence, I show you this lab trial because there I have some species in there. So in this lab test with soil and grass growing in there, 1 grub per cup, we added 400 nematodes, which is on the higher end of rates. And you can see that Steinernema scarabaei at the bottom for most of these species caused significantly higher mortality than Steinernema glazeri and Heterorhabditis bacteriophora, although these, once again, they do reasonably well against the Japanese beetle. But in scarabaei also, even against, here we have 3 Philophaga, so May June beetle species, they did very well. And just to give you even a better idea, we also have here a rate for scarabaei that's 1/8 of that, 50. And you can see against some of these species, it's just killing them just as well. And even against the May June beetles, it starts to lose a little bit, but it's clearly better than the other species. But then against mass shapers, it's for some reason they're not really any better than the other species. And against spring June beetles, none of them really does a whole lot. We also did some other studies in the greenhouse that show similar things. And then here is a summary of many field trials. And you can see once again here, we used 2 rates, 1 billion nematodes per acre, and then 0.4 billion. The data I read are the ones you saw already before for Bacteriophora. And you can see that against Japanese beetle, scarabaei is better, but it's not a huge difference because Bacteriophora does all right. But against these other species, then you can see that Bacteriophora scarabaei is far superior because it's still just as effective or almost as effective as against Japanese beetle. The other thing we looked at, recognizing that this species is rather specialized and seeing that it reproduces very well in the white grubs, we looked at long-term effects. So here this was one of 3 different experiments we did that lasted between 3 and 4 years. We did 1 initial application of nematodes in the beginning, This is putting the data together for 3 different rates, so 1 billion per acre and down to 1/8 of that, so that's pretty darn low. In other trials, we went down to a 25th of that. And in this particular trial, what you can see is that in yellow, that's the plots that were treated. There was no difference between the 3 rates because now we gave them a little bit more time. Rather than 3 weeks, we gave them 4 weeks. That allowed these nematodes to reproduce and go out, kill more grubs. Even though I wasn't expecting that, or planning for it, they wiped out every single grub after 30 days. And other trials that didn't do quite as well, maybe only 80 to 90% at the lower rates. And you compare that then with the blue line, which is the untreated plots, and you can see over 4 years, not always statistically significant, but the nematodes with the initial 1 application kept the grubs at lower rates. Although then here, over time, of course, that became more low variable. And then here in this, you can see the nematode numbers in the soil. So we took soil samples, and you can see that rather than going down in numbers right after application within the 30 days, the numbers had more than doubled, almost tripled, because clearly they had killed the grubs and reproduced them then, and then their progeny came out and killed more grubs, hence 100% control even at the lower rates. But then over time, just things became more variable. And here you can see that even on the blue line, we started to see some of those nematodes in those untreated plots. And that explains probably why in the last couple of years, there was less of a difference and no significant difference between the control and the treated areas. But yeah, so this nematode certainly can also do very well long term once it's established. But you would probably have to reapply it every couple of years. So this is not something that's going to last forever because it's going to get patchy over time. So then looking at estimating for the average Georgia life cycle here for Japanese beetles, so central, maybe it's Georgia, this would be the timings when you would ideally apply these nematodes. So late July to mid-October approximately, and ideally more maybe in second half of August into early September. But the temperatures are certainly warm enough to last that you might start to see problems already with grubs. But then Scarabee is a bit more cold adapted. So I would apply it a little later, maybe starting mid-August, but certainly through October at least. And ideally then more like late August through September, probably going to do the best job. But because it's more cool adapted, you could also apply it in spring when the grubs come up again. So probably sometime around April into May, and it would also do a good job. On summary, we saw where Cyanide Scarabee should work very well, targeting second and third instar grubs, whereas the Heteropus Bacterophora probably better only used against Japanese beetle, but that is one of the major anyway, pests. But it should target the younger stages, so start maybe from first to early third stage. And again, for grubs, of course, here we have what I said already, moisture is very important. You need to wash them in afterwards and try to keep the soil moderately moist. So that's for white grubs. Let's have a look at billbugs. Unfortunately, I have not really done much research and there's not that much out there on nematodes for billbugs, but there's some. So the 2 species that you mostly deal with probably, so it's the Bluegrass billbug, and I understand your most important one is the hunting billbug. So you're right. And this would be the life cycle. So I don't know how well explored have been so far in Georgia. Bluegrass billbug up here only has normally 1 generation, but down in the south, they should be able to maybe squeeze in a 2nd generation. They overwinter as adults, and then you start seeing larvae probably sometime imagine April, May. Whereas the hunting billbug in the south overwinters primarily as larvae, but also to some extent as adults and pupae. And then you start seeing larger number of larvae probably sometime in June or so. But there's also a difference that the hunting billbug, unlike the bluegrass billbug, can also cause problems in the adult stage. Apparently, some research from North Carolina suggests that. So there are stem borers initially, the first 3 stages, and the last 2 stages out of 5, they feed externally on the crown, roots, rhizomes. And so here is the typical, you can see when the grass dies, and you can pull it up just above the crown, the stems, and they're filled with this sawdust-like material or hollowed out, typical sign that was caused by billbugs. And that would be the kind of damage here early and then later severe damage here in spring. This is a plot with endophytic grasses, but that doesn't really work so well for you because you have warm season grasses. But maybe in northern Georgia and the foothills, you have more cool season grasses. But we're talking nematodes. So there's some old data here from the United States against bluegrass billbug, and that showed that Steinanimocarpus in particular, but also Heteroclitus bacteriophora, could provide somewhere in the 70% control, just as good as standard chemicals at the time. Of course, nowadays, some of the neonics, especially Arena, if applied at the right time, can be quite a bit more effective. And then the other species, hunting billbug, there's only data I'm aware of from Japan, where it's the biggest problem, insect problems, in my understanding, on golf courses. And before the arrival of Neonicotinoids, they didn't really have a lot of things. And so they developed Steinanimocarpus for this insect, and it was more effective than any of the chemicals had. But of course, the Japanese, as they are, like Ikebana, they really worked it out in really detail and really had the system down, and it worked quite well for them. Again, probably Carpocapsa and maybe Heteroclitus bacteriophora could be used for billbugs. And there is the species Steinanima rio brava that we have to study over the next couple of years, see if that would work. And that's a very heat tolerant species, Rio Brava Valley, obviously. So that might actually work out if they are more virulent to these weevils as well as they are against other weevil species. So for bluegrass billbug, probably when you use these nematodes, you should apply them when the larvae start appearing in the soil, which probably at New Jersey would be in mid-June, early July. So for you guys, maybe a month earlier. Whereas for hunting billbugs, it may be possible to target the adults as well as mixed populations of adults, larvae, pupae in the cooler part of the year. But you want to make sure that the soil temperature be probably at least 60 degrees Fahrenheit for the nematodes to be active. So we're talking like around April and May, maybe you can do this. And once again, they need moisture, just like for all these other ones. Now I'm going to talk about another weevil that you don't have yet, but this is the annual bluegrass weevil. Huge problem on golf courses in the Northeast and Mid-Atlantic. But if you look at here, the distribution map, this thing has now also made itself at home in the foothills of Appalachians, and it's probably even more distributed. There's no reason they shouldn't make this full line along the foothills into Northern Virginia, wherever you have enough poa and bent grass. And chances are, if you have those same conditions in the foothills in Georgia, you might end up with that one too. We have done a lot of research with that because this is a big, huge problem. Golf course here, this is the kind of damage it does. It really takes out annual bluegrass, much more so than bent grass. And people in the past have been really crazy about this insect and done 10 plus applications per year sometimes, which resulted in broad, widespread, but also broad across modes of action resistance. Overall, 20% of golf courses report to have trouble with this insect, trying to control it, which it suggests resistance. We have shown resistance in many cases. Regionally, like on Long Island, more than 55% of golf courses have trouble. And we have shown that it's pretty broad insecticide resistance against most modes of action. Life cycle, very similar to billbugs, just on a smaller scale. Stem borers when young, then feeding externally at the base of plants, but they go through 2 to 3 generations per year. Here we did some fairway trials. So you can see here along the edge of fairway, you see the damaged grass here towards the end of the spring period. And so what we did, we tested a whole bunch of commercially available nematodes here, just different species, over 3 years. And you see there's a lot of variability. So if you look at the data overall, I would say that the most consistent and reasonably good one was Steinanimocarpus in yellow, giving on average about 67% control. But that, of course, doesn't really cut it for this kind of insect that needs to be controlled at 80+% control. So we did some more trials looking at how we can improve this. What we looked at is combining them with chemicals, in particular merit, which then at the same time would also control white grubs for the year, as well as splitting the application. So applying nematodes, only half the nematodes in the beginning, and then the other half about a week later. So here the data you see now are merit. Merit doesn't really work that well. This is on the lower end, but you only get 40% control on average with merit for this insect, even when it's not resistant. Then we have here a lower and a higher rate. So that's a higher rate, actually quite as well, 1 billion and half a billion nematodes, giving us here 60% control. And here's the split application. So applied the second half, applied a week later, and that gives us a bump. And we saw that pretty consistently over several years of trials. This was on the better end, bigger, like a 10 to 20% bump. The other thing was here, when combined with merit, we also saw an additive, unfortunately not synergistic, but just additive effects. So it also gave us like another 20 or so percent. And when we did both combination with merit and split application, we ended up here with 90 to 95%, and between 85 and 95% in all the different trials. And that holds up with pretty much any chemical. And again, the merit would not necessarily be an extra application, because you would also then get white grub control from that application. So Carpocapsa certainly works quite well against this nematode. Rio Bravo we will have to test. Could be a bit more problematic than for Billbugs, because in spring when we want to control them, it's maybe a bit cool for Rio Bravo. Again, the same kind of things as far as moisture, water goes as for the other insects. Then we saw split applications as well as combination with merit control. One more insect here, black cutworm, which we did some research on. So this is a cosmopolitan around the world pest on golf course greens and tees. The larger stages, so 3rd through 6th or even 7th larval instar, spent a day in burrows, but then come up at night, feed at the surface. To Georgia latitude, you probably have between 4 and 6 generations per year, and they overwinter there. Ours up here cannot overwinter, so you send them up our way every year. Thank you very much. And here, this is a lab trial in these cups with sand, just to see how susceptible these different stages from 3rd through pupa are to all these different nematodes we tested. And we found that as far as these conditions, Carpocaps, Bacteriophora, and there's another Heteraptidus megadus, and then Steinanima Rio Bravo did very well, better than Steinanima feltia in green here. And the worst nematode was Steinanima krausei. As far as stages go, and overall, the most susceptible tended to be the 4th larval stage, with the 3rd and 5th a little less susceptible, and the least susceptible was the 6th stage as well as the pupa. And we did some other tests, but let's go straight to the field. So here are 4 tests we did. We kicked out krausei and megaliths. So what we kept feltii, because we wanted to see if under cooler conditions, like in spring or in September, maybe they would hold up. But overall, we see a lot of variability here between species. But once again, just like against ABW, the most consistent is Steinanima carpocapsae in yellow here, with an average control after 7 days at the 1 billion nematodes per acre rate of 83%. Also the most consistent with the least variability, 70 to 90%. And also the highest speed of kill at after 4 days, on average about 68%, preventing significant turf damage. But at least on greens, superintendents, this insect expect like 95% plus percent control, or at least 90, depending on the density. And so this is not good enough. So we looked into how we can improve this. And so what we did in this case, again, we looked at split application. Only now things have to go more quickly. So we applied the nematodes, half of it in the beginning, the other half after 3 days. And the other thing we looked at is the effect of syringing. So syringing is an application of small amounts of water on hot days, on greens in particular, to help the grass stay happy. And that would also help the nematodes, obviously. But we used very low rates here. So this is per hectare. So that would be 0.4 billion per acre or 0.2, because we wanted to have more space to show significant differences. Because if you get 90% control or 85, so there's not much improvement you can show. So when you see here for nematodes alone in these 2 trials here, we got up to 80% control here with that still a pretty low rate, 0.4 billion. And then combined with syringing, we got a little bit of a marginal effect of improving between 5 and 11%. And even more effective, however, was the split application, where we got improved control of about 10 to 17%. But we didn't test, which of course would make sense to do on hot days, combine both split application with syringing, you might get another 5% out of it. But if you would now do this with a higher rate, the 1 billion per acre, you would probably end up there in the 90s, and it could be competitive with the insecticides. Again, for like gutworm, the medium-sized stages were the most susceptible, and so you want to apply a little earlier, so target when they're mostly third to fourth instars. By the way, in a field trial I showed, the last one, they were a mix of third through fifth stage. And then Carpocapsid probably is the most effective species for this. And you can optimize nematodes with syringing and especially split applications, and particularly when it's hot. Okay, with that, I come to the end of my presentation. So if you want to look up some more information, you can go to my entomology webpage at Rutgers. You can also just Google my name, that will show up. And I have a bunch of extension presentations and publications on there. Or you can also email me questions or even call me. Okay, with that, I conclude. So if we have time, we should have some time for questions. Be happy to answer them.
Thank you, Albrecht. And we have plenty of time for questions. So I invite you to use the chat or unmute yourself and use that feature as well. Albrecht, I noticed that one of the first things that came to my mind was wishing that there were common names for these nematodes so that we can refer to them easier.
Yes.
Because those names can be
at the species level. Yeah, no, Heterocyanema scarabee, you could call the grub nematode, of course. Or there's a Cyanema scapteriski, which is currently not commercialized, but that would be the mole cricket nematode. But the other species like Carpocapsid, they just have a broader host range. There's no the jumper, because they can stand on their tails and even jump, but other species sometimes do that too.
That is interesting. That is very interesting. Can you talk a little bit while we're waiting for people to come up with some questions? It's fascinating to me to have something so small to infect this much larger organism. How the nematodes get into the grub? Is it by ingestion, or is it that just by contact, they just move through the skin? How does this happen?
So they're attracted by, for mostly carbon dioxide from the host, probably even movement. They can maybe feel vibrations and any other exudates from the host. And then they're mostly attracted to either the mouth, anus, or these breathing opening, the spiracles that insects have for most segments. And that's where they try to get in if they can. Of course, insects might have mechanisms to try to keep them out, especially soil insects. And they might even bore through thin parts of the cuticle, some species, like the joints of the legs or the joints of the mouth parts. And then they just eventually, if they make it through the breathing system, then they just, at some point, they get into the smaller and smaller breathing tubes and then just slash around and break it. Then they're in the body cavity. Or if they make it through mouth or anus, then at some point they bore their way through the midgut epithelium to get into the body cavity. And that's where they release the bacteria then and start havoc.
I see. Okay, so we have some a question in the chat, but before then, we have two in the questions and answers. Would you like me to read, or would you prefer to do it yourself?
Oh, no, you can read them.
Okay, the first one is, does soil type, particularly sandy versus clay soil, influence nematode efficacy?
Yes, it does. It goes both ways. So generally, coarser soil, so more sandy, is easier for them to move through. Right? There's bigger pores, especially for larger nematodes, have larger infected juveniles. But on the other hand, they need the moisture, and sandier soils, of course, tend to dry out more quickly, which could be then also limiting. Maybe some research has even suggested that for Japanese field control, the finer soils might work better because they tend to keep the nematodes closer to the surface. They don't disperse as deep, and so that's where the grubs are. Plus, obviously, the heavier, more clay content, loamy soils hold soil moisture longer. They're not as variable. So that's soil type. Okay, the cost, I see. What is the cost of these nematodes compared to mainstream insecticides? they tend to be more expensive. It depends. If you have some of the new on-patent insecticides you have to use, which you might have to use at high rates, they can cost you over $200 an acre. But if you have a cheap off-patent, old, no longer mainline product, let's say like imidacloprid, rather than merit, you might pay as little as $20 an acre. The nematodes will cost you anywhere, at least I would think $200 an acre if you use those bigger. Now this Cinnamon Rio Bravo, they sell it in large packages for the citrus growers. That is cheaper. But you have to buy 3 billion. So that would be worth 3 acres. That will cost you only about $130 per acre. So it depends on how many are used. If they can sell larger quantities, larger packages, price goes down. But yeah, Carpocapsa bacteriophora will cost you typically more than $200 an acre. So not exactly cheap. But compared to other biologicals, it's on the cheaper side, actually. Can nematodes be used for control of ticks? There has been some research, but it's not really my expertise. So they can kill ticks, apparently, but it probably depends on the tick type. I think the research was mostly with soft ticks as opposed to hard ticks. And I'm sure the efficacy wasn't very high. I think those were more trials where they tested them maybe around sites where ticks are more concentrated, like if there are cattle ticks, soft ticks, where you have a high concentration. Whereas if you would have to treat large areas along the woods edges, so I'm not aware of any research on that. Okay, I don't see the chat.
There is a question. Can you apply Dialox insecticides along with the beneficial nematodes?
Dialox was the question?
Can you apply Dialox along with the beneficial nematodes?
Okay, Dialox was one that has a bit of a limitation. We had that in the table here. Where is that table? But that would make little sense. So here's the table. Trichlorophone, which is the AI of Dialox. You should not tank mix them. You could apply them same area, but you should not tank mix them. But trichlorophone and Dialox, they both would be applied in a more curative approach. And so wouldn't really make sense to combine them, I think. Not sure if anybody has ever looked at if there is some kind of interaction. It might be like an additive effect if you apply them together.
There is another question in the chat. What's the difference in the nematodes that damage root systems in plants?
They're different species, very different species. There is at least 100,000 species of nematodes have been described. They do anything from free living, feeding on bacteria, on fungi, to parasitizing plants in different kinds of ways, to parasitizing all kinds of organisms, insects, other arthropods, vertebrates. There's all kinds of diseases caused by nematodes. They're all different species. They're as different as, let's say, a Japanese beetle is from a click beetle or from, heck, even a mite. Okay, that's a bit extreme, but let's say some beneficial insect like a ladybug, they're very different from each other. Even though they don't look as different because they're just like little tiny little worms, but to nematologists under microscope, they still look very different.
Yes, they all look like little tiny worms to me. Can you talk a little bit about one species that was isolated, that was native species? I think it was the scarabei, if I'm correct.
Yes.
Yes. Can you talk a little bit about that, how it was found and where is it native to? And
okay, so far, it has only been found in New Jersey and various areas, and then in Connecticut. Basically, it just comes down to you need to have somebody who digs up the insect hosts that get infected by them. See, people, when they look for these nematodes, usually do surveys. What they do, they take soil samples all over the place in different habitats, bring the soil back, and then throw these waxworm caterpillars in there that are very susceptible. And then if there are nematodes in there, they might get infected. But some nematodes there, like scarabei or the wall cricket nematodes, they don't really care that much about these caterpillars, and they don't reproduce necessarily as well in them. and they might fall through the cracks. So you actually have to go in the field and dig up the actual host. And when I do research if I do a lot of grub experiments in the lab or in a greenhouse in winter, we've collected up to 10,000 grubs and so that's when we come across them. Or last couple of years, I've looked for new isolates of the species and found. So we just take a whole bunch of cup cutter cores and go through them, or where we can, we take one of those small sod cutters and cut open areas that are heavily infested with grubs and look for infected grubs. And you find them; they're out there. Not only this species, other species. So that's how they're found. And yes, I'm sure if people would look harder, there's no reason why this species shouldn't be found in more areas where, similar grub species occur.
I see. We have another question that popped up. I've heard that some types of fungicides actually take away beneficial nematodes that help with fungus control. What's the best angle to approach that with turf management?
Yes, you have again the table here. So you can try to avoid using them at the same time. So here's, for example, here we have Venomil, chlorothalonil. I don't know. Try them. I don't know if that's a fungicide, but there's some fungicides here that you shouldn't tank mix. Fungicides shouldn't have too much of a negative effect on the nematodes. What you need to worry more about is typically nematicides. And in the south, of course, nematicides are pretty widely used. Nematodes, plant parasitic nematodes are a big problem. So that can be a limiting factor. Up here in the northeast, plant parasitic nematodes in turf are really a problem, and only on sand-based greens here and there. But down south, it's very different. If you get into Florida, they're like the worst. I don't know. In Georgia, I assume they're also pretty bad.
You're right, it could be. I was
But,
interested in the. I'm sorry. You wanted to say something?
oh no, go ahead.
Uh, I was thinking about the weather that we've been having here in Georgia, and we just had quite a bit of rain come in. So you mentioned how important it is to have certain level of moisture in the field in order for these applications of nematodes to have the best chance.
Yes.
So I was, and you also mentioned, if I remember correctly, that if there is very little oxygen and spaces for the insects to move, or the nematodes to move, that would not be good either. So I guess if you have in our clay soils, we could stay wet for a pretty good long time. But that's certainly something to consider, is not dry, but not too wet either.
Not too wet is less of a problem. You would have to have standing water, probably. That would be in the long term problematic. If the grass survives it, the nematodes will survive it too. It's more if it's too wet for a number of time, then it can't move as effectively. So don't overwater, but not many people are going to do that. So the dry end is more problematic typically. The soil will, over a couple of days, drain enough that the nematodes will be happy again and can move around effectively.
I see. Thank you.
So yeah, it's the heat and the water. They're limiting factors. And so for you guys, often, I don't know exactly. I assume your insects start about a month earlier and go a month longer into the fall than compared to here, depending on where in Georgia you are. And so the timings could be a little bit different. And then I don't know so much about the rainfall patterns. If you have,
We can have quite a bit at one time, and the question just came in. With too much rain over, say, a few days, could it flush the nematodes out?
No, they won't move out. That much water would mostly run off on the surface, but they should be fine with that.
Another question: can nematodes be used to control? Oh, I'm sorry, that was an old question,
Ticks.
I guess. The ticks. Yes, the ticks. I'm sorry. Now we have about 2 minutes. Any burning last questions? It was a lot of information, and I encourage everyone to go and look at the websites and resources that Albrecht put out.
Of course, most of my presentations, they're more targeted for the Northeast. We up here have a whole lot of more different white grub species, for example, Oriental beetle, Asiatic garden beetle, European chafer, masked chafers, and so on.
Yeah, we have our own sleuth of problems.
You have mostly different grasses too. You have mostly cool season, warm season. We have mostly cool season grasses up here. That, of course, makes a difference.
If there are no more questions, thank you, Albrecht. It was very informative and I really enjoyed it. I'm sure everyone learned a lot. And if you wouldn't mind hanging around till the very end, maybe if someone comes up with another question, okay. Thank you so much.